Welcome everyone to today's webcast, 2 decades in the making. My name is Cameron Fainoff and I'm the Director of Marketing and Sales here at BioSurfaces, and I will be the narrator for this webcast today. We are thrilled to have you join us as we take a deep dive into BioSurfaces' journey, our groundbreaking technologies, and the exciting future ahead. Today's webcast will cover a range of topics that highlight BioSurfaces' past, present, and future. We will begin with an overview of our company's history and mission from our president and CTO, Matthew Fainoff. This will be followed by an in-depth discussion on our three major lines of business: our in vitro research tools business, cell therapy device business, as well as our code development capabilities. We will also explore our marketing and positioning strategies and provide insights into our fundraising initiatives and future growth plans. Finally, we will close with a discussion on our manufacturing processes and regulatory strategies before wrapping up with final remarks and next steps. I'm honored to introduce today's esteemed presenters who have played a pivotal role in shaping BioSurfaces into what it is today. As mentioned, Matthew Fainoff, co-founder, president, and chief technology officer; Nathaniel Long, director of business development; Dr. Patrick Hayden, Vice President of Innovative Research; Dr. Lisa Fitzgerald, Vice President of Research; Dr. Jayashree Chakravarty, Senior Research Associate; Rayan, the Director of Device Development; Art Martin, Vice President of Manufacturing; Kyle Golden, Senior Design Engineer; and Emily Pacheco, the Director of Quality Assurance. Each of these individuals brings a wealth of expertise and experience into today's discussion, and we are eager to share our insights with you. Thank you again for joining us, and let's get started.
My wife Tina and I established BioSurfaces in 2003 to address a long-standing problem that we, and many other researchers and clinicians, face when using textile materials such as woven or knitted polyester for different medical, benchtop, and even consumer applications. In our personal experience, using these materials presents limitations such as lack of healing and scarring when used in the body, as well as limited versatility in structural design and inability to deliver active agents or biotherapeutics for different applications. Prior to forming BioSurfaces, we conducted extensive research to try to modify existing textile materials. While having some success, we could not fully address the issues when using these textile materials. BioSurfaces' focus is on developing the next generation of materials, what we call Bio-Spun, that are made from synthetic polymers that have a structure that resembles the body's natural cellular matrix. Our mission is to improve lives throughout the world by applying our unique Bio-Spun nanofiber materials paired with our decades of expertise to develop and provide a range of products that are superior to those available today. Our value proposition is that the company is a solution and material provider, allowing BioSurfaces to fully understand a pain point, followed by providing a rapid solution to the problem. At a high level, Bio-Spun materials are produced using our proprietary room temperature electrospinning process. Envision Spider-Man launching his spider web, only this is a little bit more high-tech. This web can be used for many different applications, several of which will be described later by our team. We have developed over 21 years of trade secrets to differentiate our technology from the competition. The fibers within our Bio-Spun materials are 38 to 50 times smaller than a human hair. BioSurfaces is applying its Bio-Spun materials to form three distinct areas of business: our life science benchtop tools, or what we refer to as in vitro research tools, or IVRT products, our cell therapy delivery devices, and our co-development opportunities, which we leverage our core electrospinning and product development competencies to help customers or partners with their specific applications. Within these target areas, our Bio-Spun technology has been validated by some of the world's leading organizations. BioSurfaces began as an R&D company, receiving numerous federal grants and contracts to develop specific medical devices. This allowed us to slowly grow the company on several fronts. This process, along with developing collaborations and co-development programs with outside companies, allowed the company to continuously expand the Bio-Spun platform. This strategy, which provided non-dilutive capital, allowed the company to develop various applications such as hemodialysis access and peripheral vascular grafts, a cell therapy delivery device, sewing cuffs, a perianal fistula plug, and a dermal substitute, just to name a few. Each device has an extensive data set which can be licensed by a customer or co-developed with a partner. Within the past 10 years, BioSurfaces began to apply its BioSpun scaffold onto plastic housing that can be used to produce various types of tissue. This was the start of the In Vitro Research Tools, or IVRT, product line, which does not require FDA approval for use. In addition, BioSurfaces also broadened its non-regulatory product offerings when approached by a global cosmetic company interested in using its BioSpun scaffold to deliver actives directly to the skin. BioSurfaces continues to focus on growing its IVRT product portfolio, either selling directly to customers or through partnering with other benchtop product companies. We are expanding the use of our BioSpun materials for cell therapy delivery, as well as aggressively pursuing more co-development business for various applications. The company is generating revenue from all three business lines. This year is shaping up to be one of our busiest years. Customers and outside companies are starting to realize the potential of our BioSpun materials. BioSurfaces is at an inflection point. Our immediate challenge is to be able to meet the projected demand for our BioSpun technology. This will require the company to grow our team as well as to expand our footprint in order to increase manufacturing capacity from pilot scale where we are now to full-scale manufacturing. BioSurfaces made a strategic decision to bring in outside investment to help tackle this challenge. The company was fortunate not to need any outside investment for many years due to having various ways to generate revenue, which allowed us to develop the technology platform while responsibly growing the team. We are currently raising a Series A round that is designed to tackle the trials that growth brings with it. We are making great progress and continue to look for investors who believe in our mission. It has been a challenging environment to raise capital, but we remain undeterred as we know our BioSpun material has the potential to impact many different areas.
All of the materials, scaffolds, and devices we create here at BioSurfaces is through our electrospinning process. The result of our electrospinning approach gives us what we call our BioSpun products. Throughout the company's 21-year history, we have iterated and perfected our approach with multiple different types of polymers. Some of these BioSpun products can be seen in our in vitro research tools, or IVRT as we like to call it, where we take our flat sheets and attach them to various individual inserts like 6, 12, and 24 well plates, onto 24 and 96 well high throughput screening plates, or onto microphysiological system devices like what we have done with FinAdvance's microfluidic plate. This is one of our main lines of business where we were trying to give researchers a better benchtop tool with our 3D animal-free BioSpun scaffold. BioSurfaces took a backwards approach when it came to the IVRT business. Throughout the first 15 plus years of the company, BioSurfaces created medical devices that were tested in vivo, which generated a lot of positive data to help both us and our customers understand what the BioSpun scaffolds brought to the industry. A few years back, we realized that we could apply our scaffolds to plastic housing units of various sizes and use those for cell culture testing in vitro. With the knowledge that we already had, we knew that the cells would grow into our scaffolds since they have already done so previously in vivo. We know that our BioSpun scaffolds can translate from in vitro to in vivo because we have gone from in vivo to in vitro. The cell culture industry is an estimated $16.59 billion market with many different players. The market using 3D and 3D microphysiological systems for cell culture is currently being valued annually around $2 billion and $294 million respectively, with compound growth rates of about 11.22% and 33% respectively. There are many different competitors and competitive products in this space, ranging from 2D scaffolds and hydrogels to organ-on-a-chip and developed tissue model systems. The list of these can seem endless, so I won't dive into them, but it is worth noting that some of these competitors in the space have worked with us and are our partners. When it comes to targeting markets, we try to take a niche approach and find customers who are looking to dive deep into our product and utilize it for what it does best, which is its 3D extracellular matrix components that mimics the human body's extracellular matrix. We have found success in reaching out to pharma companies who can test our BioSpun scaffolds on the benchtop and potentially move into a product discussion with us. Other targets for us have been cosmetic companies who like to build partial or full thickness models with our scaffolds. These are just a couple of major target markets that we have looked into.
Well, one of the recent things we've introduced is utilization of our electrospun scaffold products for in vitro or preclinical. screening of electrospun materials that are ultimately intended for use in medical devices. Specific examples where this approach has been highly beneficial include screening and optimizing materials for our current SBIR project that involves development of a pancreatic islet cell chamber for type 1 diabetes therapy, and also our Gates Foundation project that involves development of implantable cell chambers for therapeutic antibody production. Now, these are topics that Lisa will elaborate further on later in this webinar. So using this approach, we're able to find the best material for a particular cell or tissue type on the benchtop before moving forward with animal or clinical studies. This reduces costs and time and avoids unnecessary animal experiments. A similar approach has been successfully utilized by several of our partners and customers in their own clinical development programs. A second major focus is on development of 3D in vitro tissue models that may be used for preclinical toxicology and drug development applications. An active internal phase 2 SBIR project is currently developing advanced human full-thickness skin and airway tissue models using completely non-animal derived cells and media formulations. Our aim is to provide turnkey protocols to allow researchers to use our scaffold products to produce these human tissue models at scale in their own laboratories. These products help avoid animal use and provide better translation of results to in vivo human clinical outcomes. Numerous other full-thickness human tissue models, such as intestine, ocular, kidney, liver, and others, will also benefit from the use of our 3D scaffold inserts. Models developed using our in vitro research tool products may also have application beyond in vitro testing. For example, our in vitro human skin models have potential for in vivo clinical applications in areas such as skin grafts for burn patients or diabetic skin ulcers. Ocular models may have potential for in vivo translation for diseases such as macular degeneration. One specific area for concern for both in vitro and especially for potential clinical applications relates to regulatory approval and compliance. To address this, we have established a robust quality management system to comply with GMP and ISO 9001 and 13485 standards and federal regulations for manufacture of medical device materials. Another area of focus relates to regulatory acceptance of in vitro tissue models and protocols for use in toxicology testing applications. As a part of our ongoing SBIR project, we are characterizing the reproducibility of skin and airway models and validating them for specific regulatory applications, such as screening for skin irritation, phototoxicity, corrosion, and sensitization potential.
The future of BioSpun scaffolds is incredibly promising, and we're excited about the potential applications across various industries. In regenerative medicine, we're looking at opportunities for personalized tissue engineering, such as more patient-specific scaffolds to repair damaged tissues. There's also significant potential in advanced wound healing, where our scaffolds can serve as bioactive dressings to accelerate healing and reduce scarring. Another key focus is in drug discovery and testing. BioSpun scaffolds are perfect for creating more physiologically relevant tissue models for high-throughput drug screening and organ-specific toxicity testing. This can help pharmaceutical researchers get more accurate data earlier in the development process. We are also exploring how this technology can enhance cell therapies, providing a consistent animal-free platform for stem cell cultivation and even gene therapy applications. We are also making strides in advancing the 3D tissue models for disease research, whether it's for studying cancer, neurodegenerative disorders. These models can provide a reliable tool for researchers working to understand complex diseases and develop better treatments. The cosmetic industry is another area we are focused on, particularly in expanding our skin models for non-animal testing and potentially for dermatological therapies. Longer term, we want to continue collaborating with researchers and industry leaders to unlock new applications for our BioSpun scaffolds. So whether it's advancing drug discovery, regenerative medicine, or cosmetic science, BioSpun is positioned to make a meaningful impact in a variety of industry, and we are just getting started.
BioSurfaces has continued to make strides with its cell therapy devices, especially over the last few years. We focus on two different products in the cell therapy space, the BioSurfaces cell chamber and the BioSurfaces cell therapy scaffold. The cell chamber has had a few different iterations over the years thanks to the hard work that Rayan and the rest of the team has put into it. We offer a few different sizes of the chambers. One in particular is a mini cell chamber that can be implanted in the subdural space in the brain. Our engineers and scientists have exceptional skills when it comes to iterating these devices to help customers with their problems. We know that our device does not induce fibrosis when implanted and allows for nutrients to flow into the device while allowing therapeutics to flow out.
We're currently developing and testing in-house two different implantable cell encapsulation devices, or cell chambers, to address specific medical needs. The first is a chamber designed to house insulin-producing human donor pancreatic islet cells as a potential treatment for type 1 diabetes. Islet transplantation is not new, but patients currently receiving non-modified donor islets must also be placed on potent immunosuppressive drugs for the rest of their lives in order to prevent their body's immune systems from rejecting and attacking the islets. The walls of our cell chambers are specifically designed with multiple porous layers, including one which will prevent immune cells from getting in and from the cells inside from escaping, all the while allowing nutrients such as glucose and cellular product, in this case insulin, to cross through that barrier. Another clinical application for our cell chambers is to provide housing and immunological protection for cells genetically modified to produce a particular drug. Once implanted in a patient, the cell-loaded chamber will serve as a drug factory within the body. This will allow for more long-term, steady state levels of the drug inside the patient and eliminate the need for repeated dosing by pill, injection, or infusion for as long as the cells remain viable, at which point the cell chamber will be removed and replaced. We're currently developing a chamber to house cells modified to produce anti-HIV antibodies, with the goal of providing a year-long treatment for a patient with just one simple insertion procedure. We've been conducting preclinical testing of our cell chambers using both in vitro cell culture and animal models for several years and have gotten some very promising results. Through our prior R&D collaboration with Takeda Pharmaceuticals, drug-producing cells in our chamber remain viable when implanted subcutaneously in a mouse model with continued drug release detectable in the plasma for over 14 months. In another study, cells loaded into our Bio-Spun mini chamber and implanted into the subdural space of a rat model showed drug detectable in the surrounding tissues of the brain and in the rat plasma for 50 days. This approach may allow for drugs targeting brain cancer or other diseases of the brain to actually cross the blood-brain barrier, which is an ongoing challenge. For our anti-HIV project, which is funded through the Gates Foundation, we're currently testing drug-producing cells on various types of Bio-Spun materials within our IVRT plates, that's our in vitro research tools, to optimize the cell chamber layer configuration. We have the ability to design the interior as well as the exterior of our chambers, and different cell types may prefer or grow well or function better on different types of electrospun material. So this gives us a chance to optimize what the cells like to grow on before we construct the chamber specifically for that cell type. In our current human islet project, we've thus far demonstrated islet viability and glucose responsiveness, that is insulin production, for up to 50 days in culture. Implant studies in a diabetic rat model are currently underway with preliminary data thus far showing the islet survival and function in an immune-competent rat after explant following a 14-day implantation of one of our cell chambers.
There is definitely a need for our cell therapy devices for treating chronic illnesses such as diabetes, liver disease, and other rare and infectious diseases that require the long-term delivery of protein drugs or biologics. For example, we are working on a grant from the NIDDK to encapsulate islet cells into our cell chamber for the treatment of type 1 diabetes. We're also working with the Gates Foundation to create a device that can provide long-term delivery of a therapeutic agent for patients in third world countries. Another company is using our cell therapy scaffold for hypoallergenic cells, and many other projects are ongoing. So we do see the need in the market for our cell therapy devices, and we are actively working on fulfilling. In terms of upcoming innovations, we are constantly working on improving our devices and coming up with next-gen ideas. Currently, we are working on a couple modifications for our cell chamber. We're developing a method where the cell chamber would have a port that would reseal on its own, since right now we are using an ultrasonic welding process to seal the chamber after cell loading. So a resealable port would eliminate the need for that. Another modification is creating more surface area inside the cell chamber to give more room for the cells to grow. And we're constantly modifying our cell delivery devices to fit our customers' needs. For example, if a company we are working with needs a scaffold that is thinner or thicker, or a cell chamber that is a different size or a different shape, we can work with them to make those changes. And I think that that is also another advantage of our cell therapy devices, is that they can be modified in those ways.
BioSurfaces is positioned at the intersection of biotechnology and medical technology, offering cutting-edge solutions through our proprietary Bio-Spun technology. We differentiate ourselves by developing innovative scaffolds and cell therapy devices that address critical gaps in research and medical applications. Our in vitro research tools are the first scaffolds proven in vivo to demonstrate material capability for cell integration, setting us apart from conventional alternatives. With a strong foundation in both research and commercialization, BioSurfaces bridges the gap between advanced material science and real-world biomedical applications. At BioSurfaces, we take a multifaceted approach to marketing, ensuring our brand remains at the forefront of biotechnology and medical technology advancements. Our presence at key industry events such as SOT 2025, the Society for Biomaterials Annual Meeting, and the HERN 10-Year Symposium allows us to showcase our latest innovations, including our groundbreaking research on type 1 diabetes. Additionally, our content marketing strategy plays a crucial role in reinforcing our industry expertise through blog posts, social media updates, and thought leadership pieces, such as the recent spotlight on Dr. Jayashree presentation at EBITDA. Our strategic partnerships, including the Gates Foundation funded initiative, further strengthen our credibility and visibility in the field. Our marketing efforts are directed towards three key market segments. We actively engage with biotech and pharmaceutical research companies that require advanced in vitro research tools for drug discovery and tissue engineering applications. We also work closely with medical device and cell therapy companies that seek biomaterial solutions to enhance therapeutic outcomes. Additionally, we collaborate with academic and government research institutions that conduct biomedical research and can benefit from our Bio-Spun technology. Marketing is a key driver of BioSurfaces' growth strategy, supporting our expansion through a variety of initiatives. We continuously enhance our industry presence by participating in high-profile conferences and publications to establish thought leadership. Our strategic outreach and educational efforts involve engaging with potential customers through webinars, webcasts such as this, and scientific collaborations. Strengthening our partnerships with distributors and research institutions helps increase adoption and credibility of our technology. Additionally, we leverage targeted digital marketing, including search engine optimization, email marketing, and social media campaigns to expand our visibility and reach to a broader audience. Through these strategies, BioSurfaces ensures sustained growth and reinforces its leadership position in biotech and medtech markets.
So BioSurfaces has historically been a research-based company. In the more recent years, we've gravitated to our hybrid company, where our research has led us to develop some pretty innovative products. With that, we've assembled a great team, a talented team, that's at work validating the manufacturing processes for those products. And as I mentioned, our talented team is busy at work validating our Bio-Spun scaffold product line. I think it's best to allow our department leads to explain the work that they are doing toward that end.
I would say that we're a department that has evolved into being capable of developing equipment that can be used in a validated process. Shortly after I came on, we began to work on a machine that produces three-dimensional electrospun scaffolds. These scaffolds were intended for use in vitro research tools, but also a wide range of uses in cell chambers, cuffs, and direct shipment to companies. We've worked to validate the process so that we can ensure that we're providing a reliable and repeatable product to our customers.
So BioSurfaces has fully implemented an eQMS, and we continue to put the system to use. Our current focus is to establish manufacturing work cells, which encapsulate every aspect of our manufacturing processes. So for example, we have a work cell for creating polymer solutions, another for electrospinning the scaffolds, and eventually we'll introduce more work cells that are specific to applications like IVRT and cell delivery. Establishing and validating these work cells will ensure that all processes are working properly, and most importantly, that every BioSpun scaffold meets the defined specifications. So BioSurfaces is working towards our ISO 13485 certification with the hope of being certified within the next year. Our eQMS has been structured and implemented to be ISO 13485 compliant, so we believe this goal is very achievable.
As we wrap up today's webcast, I want to extend a sincere thank you to all of our presenters for sharing their expertise and insights. A special thank you to Matthew for taking us through BioSurface's history and vision, helping us understand how far we've come and where we are heading. Thank you to Nate for outlining the impact of our in vitro research tools business and how we are positioning BioSpun for success. Pat and Jayashree, we truly appreciate your updates on our latest testing and research advancements, giving us a glimpse into the future of our technology. Lisa and Rayan, thank you for providing an in-depth look at our cell therapy device section and the exciting innovations taking shape in this space. Art and Kyle, your insights into our manufacturing processes and equipment development have highlighted the complexity and innovation behind what we do. And finally, Emily, thank you for sharing the progress and future goals of our regulatory efforts, ensuring that BioSurfaces continues to meet industry standards and expand into new markets. And lastly, to all the people listening and joining us today, thank you for stopping by. Your support and interest in BioSurfaces is truly what drives us forward. If you have any follow-up questions or would like to connect further, please feel free to reach out to me at cameron.fainoff@biosurfaces.us. Or if you want to check us out on our website, it's biosurfaces.us. We look forward to continuing this journey with all of you. Thank you again and have a great day.